we're happy to have Elias. Elias is finishing his Ph.D. This is the graduate seminar. Elias did a lot of work in both computational imaging and uncertainty quantification under the joint supervision of Yoav, who is currently in Zoom from the US, and myself. And he prepared a lot of slides, so be focused.
Okay, so hi everyone. So my name is Elias, jointly supervised PhD student under supervision of Tomer and Yoav. And I'll be telling you about 3 different ways we can use deep learning in computational imaging. But by the way, note that these names here in the slides are actual experimental measurements in a fluorescent microscope. So these are point sources. I'm not going to tell you how. Okay. So before we talk about how deep learning can help, let's first understand what's computational imaging. So the idea is that you have some scene that you want to image, and this scene goes through some sensor, for example, having some optical elements such as a lens or something else. And then the resulting measurements go into a reconstruction algorithm that tries to basically recover some property of the scene. So it could be, for example, depth estimation. And so the idea is that if we now replace this algorithm with a deep neural network, we recover using the measurements, we recover the properties using a deep neural network. This enables us some unique abilities. So in the first part, I will talk about how this thing jointly with a task-driven reconstruction loss, we can basically optimize the sensing and the reconstruction algorithm jointly end-to-end. This will be part 1. And in part 2, I will also tell you how we can visualize and quantify uncertainty using deep learning. And in both parts, I will focus mainly on bioimaging applications, but in the second part, this actually applies also to any different type of images, as I will show you later. Okay, so let's start. So in part 1, our main field of application was localization microscopy, or in its other name, palm or storm. And this is a super resolution technique that basically tries to bypass the optical limit or Abbe's optical diffraction limit on imaging a microscope. So for example, you see here on the left an image of microtubules image in a microscope, which are these like filament structures. And you can see that biology basically happens at the nanoscale. But the best we can do in a standard microscope is image something with a resolution of up to 250 nanometers. So the question is, how can we get a super resolution or super resolved version of this structure? And there are many techniques. The one we focus on is localization microscopy, and the idea is the following. So broadly speaking, we want to separate space over time. So instead of capturing a single image, we capture a movie where we attach fluorescent point sources to the sample at hand, and we blink only part of these fluorescent tags in each frame. So we capture a movie. And in each frame, we take each spot and fit it using, for example, a Gaussian fit to get the center and put it in a super resolved reconstruction in the computer. And if we repeat this for many, many frames, then we end up in the computer with some super resolved version of the same sample we started with. And roughly, this is a 10-fold order in resolution improvement. Okay, so obviously this idea wasn't ours. It won its inventors a Nobel Prize back in 2014. And basically, what we're interested in this talk is to talk about how can we leverage this method for 3D structures, because obviously biology is 3-dimensional, and there are many ways to extend this to 3D. The one we will talk about is called PSF engineering, and to understand it better, let's focus on a single point source moving in a microscope. So here on the left, you see a single point source moving in Z in the axis direction, and on the right, you see the camera image of this point source being recorded, which is the PSF. And basically, the idea in PSF engineering is to remedy some of the flaws of standard microscopes, which are twofold. One, you see that the PSF, the shape is symmetric above and below focus, so you can't tell if it's above or below. And another thing you see is that the Z range or the effective Z range is very narrow. So after going out of focus, very rapidly the signal is lost, and we're drowned with noise. Okay, so the idea is to manipulate the Fourier plane, which is basically here, by inserting an additional optical element called the phase mask. Now, once we insert this phase mask into the system, what we get is a PSF that is not symmetric above and below focus, so the shape is different, and you see that also preserves the signal nicely across very broad Z range. Specifically, this PSF is called the tetrapod PSF, and was shown to be optimal for estimating the 3D position of a single point source. Now, using this trick, how can we extend the previous approach I showed you to 3D? Well, it's rather simple. You insert the space method into your optical system. You measure again a movie of blinking point sources attached to your sample. And now, using some algorithm or some model of your shapes, you can super resolve or reconstruct a 3D point cloud or a 3D volume. Now, here you see that is encoded in color. And this seems like perfect, right? What's left to be done then? Well, the idea is that reconstructing this volume alone took about 1 hour, right? Why is it so long? And the idea is basically merely algorithmic, right? The challenge is merely algorithmic. Fitting sparse emitters you can do easily. If it's a single point source, you can fit it. But if you have lots of them and they're overlapping on the sensor, it becomes much more challenging. So basically, why is it slow? Because we need to acquire many, many frames. In fact, somewhere around 100,000 frames in order to reconstruct a single 3D volume. And so the idea we had, or that people had and we extended, is to acquire basically much less frames, but in each frame acquire much more point sources, right? So they're not going to be sparse, they're going to be slightly dense. The shapes will overlap, but then we will get a lower number of frames and we can reconstruct it much faster. But this will necessitate a reconstruction algorithm. Okay, so in the first part, I will tell you about 2 works that we did. The first one was published in Nature Methods 2020, and the follow-up, which is almost a better solution, a year after in PPAMI. I'll just mention that in our works, we started this series of works back in 2018, where also in 2D, without even extension to 3D, the 2D problem of localizing nearby point sources is also very challenging. But obviously, I'm not going to go into this because we don't have time. Okay, so moving on to the challenging task of 3D reconstruction. So the idea that we had in the first work was basically fairly simple now. We wanted to take a 2D image of overlapping PSFs or point sources images and feed this into some reconstruction network, a CNN in this case, that takes this 2D image and outputs 3D positions of the emitters in this 2D image. Now, specifically in our implementation, we implemented this with an occupancy grid. So the output is a 3D occupancy grid where in each voxel, it's occupied voxel stands for emitters, for localizations. And the rest should be 0. Okay, so how do we train this thing? Well, we have a loss function that's a combination of 2 terms, one that encourages very accurate 3D localization, and one that encourages detecting as many emitters as we can, increasing the detection rate. Okay, so testing this on simulation, you see here in black that our method is much better than the competition in detection rate, where higher is better, quantified in this case by the Jacquard index as a function of the density. And also you can see that our method is better in precision, where in this case it's quantified by RMC, so lower is better in this case. And in both cases, the black line, which is our method, is much better than the competition at the time. And just to give you a hint of what this density axis really means, what if we take an example, a visual example at a density of roughly 0.1, you see that in this case, our method recovers the underlying ground truth point cloud much better than the competition. Okay, so this is all nice, right? How did we train the CNN? We trained on simulations. We have some optical model of the system. We tested on simulations, we beat the competition, but does this translate to real data, right? That's the real question. So to answer this question, we performed an experiment where we labeled mitochondria, which are these like tubular structures in cells, and we imaged them. So this is one example frame from this experiment. And now I'm showing you the rendered frame of the recovered 3D positions of that experimental frame by the network, and again, the experimental frame for reference. If I now flicker between a couple of these, you see that our network is recovering most of the emitters very accurately in almost all frames. Some are being lost due to SNR, but the majority are being reconstructed. And if I now take all of these recovered 3D positions and render them again into a 3D point cloud or a volume, we get something that looks like this. And as you can see, this result, which we'll examine in a second, is rather nice. Again, color is encoding depth here. And what's really important is that this volume was acquired with 5 to 10 minutes, right? So roughly a factor of 5 to 10 in temporal resolution improved. Now, you might think that this comes at the cost of precision, right? We acquired less frames, we have less information, probably less accurate. And actually, that was not the case. So if we now, for example, examine this one in a relatively dense area, and we take an XZ cross-section, we can recover the theoretical tube-like structures of mitochondria as we expect from other imaging modalities. Okay, so that was in a nutshell the first one. So moving on, we ask ourselves, can we do even better? Why would we think we can do better? Well, if you look at this example frame, you see that this shape is very large. Right? It's easily overlapping on the sensor in XY. And actually, if we come back to what I told you a couple of slides ago, this shape was optimized to be the optimal one for sparse emitters, where you image only a single point source. But what we're really asking now is what's the optimal PSF for high density imaging, right? Knowing that we will have overlaps or nearby point sources. And so to answer this question, we came back to the drawing board. So thus far, what did we do? We had a simulation of a 3D point cloud, passed it through our optical system numerical model, and simulated 2D images, which we then fed into a reconstruction CNN that tried to recover the point cloud. And we did improve performance, but that there was still some points being missed. So now what we're asking really is, what's the optimal phase mask to put in Fourier plane such that the 2D resulting 2D images, when fed into a reconstruction network, will do the job perfectly. And if you can look at the structure, it sort of like resembles an autoencoder, those of you who come from computer science. And basically, we can optimize this phase mask now that we have an end-to-end differentiable system simply by backpropagation, right? We can let the net decide the optimal phase mask that it wants to receive in order to process the measurements the most easy. Okay, and this is the optimization steps that I'm showing you here. So you see the phase mask being updated in the Fourier plane and 5 axial slices of the PSF as a function of Z. And what you see is that basically it starts to optimize fairly in the middle of the axial range at first, and then slowly it also recovers the end parts, optimizing the entire range. So now if we come back to that simulation of a point source moving in a microscope, insert this learned phase mask into the system, we get the following PSF. And as you can see, this PSF is much smaller than the shape we had before, which is very intuitive because we're expecting to image nearby points. So we want the shape to be small, and then it will less overlap, less likely to overlap. And in fact, one more funny observation about this phase mask is that it actually looks like a Holmes plate to me, which quite interesting bias of the algorithm. Okay, so if we test now in simulation, how is this doing compared to a reconstruction network with a fixed optics of before, the typical PSF? We see that we do much better in detection in this orange line. And if we take 2 example images at different densities, you can actually see why. It's really exactly the intuition we had before. It's simply a smaller shape, and therefore it less overlaps. You can squeeze in much more points before it starts to overlap critically. So does this come at a cost of precision, right? We're shrinking the measurement, less pixels. You might expect that this hinders your 3D localization ability. Actually, no. Also in precision, we do better both laterally and axially. And again, RMC measures precision, so lower is better. Okay, so that was the second one. Now moving on to a very interesting application different from the one we started with, which is fast 3D tracking of DNA. So interesting application in biology besides super resolved 3D reconstructions is to track telomeres, which are these tips of chromatin, of chromosomes inside the nucleus of our cells, right? They are just the edge points of the nucleus, of the DNA inside the nucleus. Now, why is it interesting to image these? Turns out that their subcellular diffusion behavior, their diffusion coefficients, and their dynamics inside the cells tells us a lot, or tells researchers a lot, me not so much, about sleep and cancer research, with several applications that interest in this field. And so naturally, these things are in 3D, in a 3D volume, which is the nucleus, so they're embedded somehow in 3D space. But again, in this case, we can only image in 2D, right, in microscopes. So here you really see that our method is critical, right, because now things are not attached to some sample and blinking on and off, but the sample is static. Now the sample itself is moving, and they're not blinking. They're constantly on, but a lot of them are out of focus because of their depth, right? So the standard PSF just diffuses out of the image, and you don't see them anymore. And so if you want to accurately recover all of these, you need somehow to measure in 3D in each frame, which is exactly what our method does. So with this learned PSF that I showed you a couple of slides ago, we were able to track these telomeres and recover the 3D tracks in a live mouse cell in this case. And in fact, this wasn't only a proof of concept. This turned out to be something interesting. So we had a follow-up work, which is almost purely biological, using the method. And in this case, we were able to characterize the role of a certain protein, lamin A, in this case called lamin A, along the different phases of the cell cycle. So in different parts or different times of the gating of the cells. Right, so moving on to our last part to our last bullet in part one, we extended this approach to multiple sensors. Right, so what does this really mean? So I've told you already that we can increase the detection rate from this tetrapod baseline with a reconstruction network to this end-to-end optimized system, and it improves a lot in detection, but still people came to us with various applications such as nanoparticle stuff in several settings where they needed higher detection rates. So the question we're basically asking is, can we increase detection rates even higher at high density? Can we somehow still optimize the system a step further? And the idea we had basically in this follow-up work was to, instead of capturing 100% of the signal in a single imaging channel, what if we give up on some SNR and we tunnel the intensity between two channels, tunneling only 50% to one imaging channel and the other 50% to a second one employing a different PSF. Right, so now we have two images with strictly reduced SNR, but more sensor area. Therefore, maybe we can use this additional camera to somehow improve the detection rates. So before I tell you what was the result, obviously given two images, we need to somehow reconstruct, and in this case, we simply concatenate them and feed them to the same reconstruction network I showed you from earlier. And why is this important? Because this basically disentangles the performance boost given by the network and by the measurements, right? So since it's the same reconstruction algorithm, it's going to be, any improvement that we get is going to be solely because of the input measurements. So what's the optimal PSF that results in this case? How should we design it? Well, you already probably have guessed. We can simply learn it end-to-end with the reconstruction network, right? Two PSFs in this case. And this is what we get. So we get at first like shapes sort of revolving very fast around, and it's hard to interpret. But if you overlay them, if you look at the different channels in different colors and overlay in the same image, you see that basically they're somehow cooperating. Right? There's some coordinated side lobe movement that is encoding the 3D position in some very convoluted way. So how does this compare to the single channel study that I showed you earlier? So this was the detection rates we got from before, and this is the new system with two channels learned end to end. And as you can see, it's much better in detection. And also in precision, again, measured by RMC, it was much better. And obviously, simulations are simulations. The complicated the system gets, you need to somehow implement it. So there's always that fear that this doesn't translate to reality. And in this case, again, we imaged live telomeres in a single cell nucleus and track them over time in 3D. So here you see the captured images and the estimated nuclear envelope of the nucleus of the cell, the localized telomeres and the tracking time bar. If I now play this movie, you can see that basically we recover almost all of them with various different motion types across the entire volume of the nucleus. So to further validate this result that we're not just spitting out 3D tracks, we took the 3D positions that the network recovered and re-rendered the 2D measurements and compared them to the experimental data. So here you see the two experimental channels. On the top right of each, you see the measured zoom in the white square, and below it the recovered zoom in, which ideally should be the same. If I now scan this square across the entire volume of the nucleus, you see that we're recovering basically the shapes, a denoised version of the shapes very well across the entire field of view. Okay, so summary so far, because I've been really fast about this. So we can learn a model that does 3D localization of dense PSFs very efficiently, completely in simulations and translate into experimental data, which is really nice. We don't need to worry about data maintenance. We can also learn the optimal optics, as I've shown you, on one sensor, and performs even better than just learning a reconstruction network. And furthermore, we can do this with multiple sensors, and it turns out we can do even better if you design two cameras jointly with a reconstruction network. So back to the chart that we had from earlier with the applications that were somewhere up here. Now with this system, there's exciting new opportunities if you're a biologist in this field. Okay, so that was in a nutshell part one. Before I move on to part two, one last thing related to part one. So after this work, we actually had an idea. It was roughly around time COVID came around, that basically we wanted to combine this technique with a drug screening or high throughput microscopy. Right? So what do you need to do in drug screening? You need to measure tens and hundreds and thousands of samples very, very quickly. I wondered whether this technique of PSF engineering can somehow improve the performance of such systems. So very briefly, I'm not going to go into this topic in detail, but I'll just say that it wasn't actually like a trivial extension. Turns out this is really complicated, and we had a patent about it even, and some paper recently accepted to Nature Communications. And this is a joint work with the company that makes these high throughput microscopes called Sartorius in this case. Okay, so at a very high level, what does this really look like? So basically, we took this phase mask that I was telling you about earlier, and we plugged it back into an objective. So we basically almost glued it at the bottom. And now using this phase mask, which I will show you, I'll pass around in a second, we can scan with this system very large number of samples very, very quickly. And this basically enables all sorts of applications that benefit people in drug discovery. For example, one such application is going from a snapshot image to a 3D cell segmentation. Basically, from a single image, we cover the segmentation of all 3D cells inside an image. And actually, this was a really nice result that we didn't expect we would get. Okay, so I'm done with part one. I'll be moving on to part two. Any questions so far? Okay, looks good. So in part two, I will talk about two works. One recent work we got that got accepted in our RIPS, and one even more recent work that we just submitted to HCV. And before I move on with these works, I'll just say that we had also a sandwich method somewhere in between with Omer, who just finished his PhD a couple of months ago. But obviously, due to the interest of time, and it's not my method, I'll not talk about this. Okay, so why uncertainty quantification? So as I've already shown you, nowadays, these deep neural networks are almost everywhere. They're almost an indispensable layer in this field of bioimaging, right? So in 3D localization, 3D particle tracking, and some other stuff that I didn't even show you, where we go and try to super resolve images in 2D. And obviously many, many more applications, MRI or whatever other applications you might have in mind. And this is actually somewhat problematic, right? Because more recently, even us, we tried to do some sort of scientific discovery or scientific conclusions based on the outputs of these models. And this is when, you know, your method, you want it to be used, but sometimes you start getting cold sweats when people start using it around the world, because this is really wrong, right? These models are known to fail in unexpected ways, catastrophic failures. So we need to somehow quantify the uncertainty of what's going on here before we report something that should somehow guide the scientific community forward. So to go into this field of uncertainty, let us slightly step outside of the bioimaging one that I showed you earlier and look slightly on image restoration, recent advancements in image restoration. So the idea is that, assuming we have this task of painting the eye area, and this is the input image, we will call this Y. And we will assume that this image was generated by some source image that we don't know, which is called X. And we want to predict some prediction given this Y, which we will call X hat. Now, obviously, we would like X hat to be very close to X, but in such post-inverse problems, there could be many X hats that basically would correspond with this measurement. But before I tell you, or before I show you how these different X hats look like, let's first look at the statistical setting we're looking at here. So formally speaking, we assume X comes from some prior distribution P of X, and we assume that Y is generated from X by some unknown mechanism P of Y given X. Right? And this could be stochastic, this could be just masking, any mechanism. And now, basically what we're asking is, what are the different options X hat that could comply with Y, or what's the uncertainty in X hat given Y? And this is encapsulated in the posterior distribution P of X given Y. right? And so obviously there could be many x's that correspond to this y. And the recent idea in image restoration was, okay, if there's many options, simply sample, right? So you have a bunch of samples, and you can report this to the user as some depiction of uncertainty. And this is actually proposed in many recent works in recent conferences. Now, this all seems nice, but posterior sampling has many drawbacks. The first one is, well, we show in the paper, I'm not going to show you quantitative stuff because we don't have time, but we show in the paper that it could be actually, for state-of-the-art ones, it could be slightly inaccurate. So it's not really sampling from the posterior. That's one. And more importantly, to produce every single sample here, you need a lot of compute, right? So for recent fast state-of-the-art samplers, you need somewhere around 100 neural function evaluations. So 100 times passing through a neural network to produce one sample. And if you really want to visualize uncertainty, then this requires many samples, right? The user needs to sample a lot of samples, and then afterwards to look at these samples, somehow eyeball all the different options, which is very tedious and time-consuming. Okay, so you might say, okay, you have a lot of samples, let's summarize them then somehow, instead of this prolonged eyeballing experiment. Okay, that's a good idea. So how would you summarize the samples? So the common approach is to take only the posterior mean, right? Average all of them, which is a good idea, but remember that this actually discards all uncertainty. So you're averaging everything out, you don't know the different options now. So in order to complement this information, what people usually did, or up until recently did, was to report some per pixel heat map. For example, the standard deviation per pixel that somehow depicts the variation of different pixels in the image. And this was proposed in many different sorts of forms, for example, using Gaussian fitting per pixel, confidence intervals per pixel, and many more. But as you can see, this per pixel heat map doesn't tell us much about the underlying option, right? So it's of limited information in this case. It just tells you that it's certain in the skin color and everything else is just uncertain. But doesn't tell us how uncertain. Okay, so a better idea in this case is to replace this per pixel heat map with the principal components, right? To do PCA on top of the samples. Now this is very informative because now you can take the principal components and scan around the mean along each of them and see the different options. See how the reconstruction might have changed. So for example, the eyebrows changing up or down, or in this case, somehow the darkness around the eyes. So this is very nice. But really, what we really want to do is to make all of this very fast, right? Because this step here could take a lot of time, hours sometimes. So if it takes that much time, probably most people won't use it. And this is exactly what we proposed in our first work from last NITS. We want to basically circumvent the sampling step, right? Neglect it completely, and instead learn a neural network that goes directly from the input measurement in a single forward pass and predicts the top principal components and the posterior mean for any input image. Now, how do we do this? Here I'm going to show you the simplified version of our method, where we train, assuming we have some pre-trained conditional or pre-trained model that was trained to minimize the MSE and therefore outputs the posterior mean, right? But in our full work, we also have a version where we output both the mean and the principal components in a single model. Okay, but I will show you now with two models. So starting from this MSE minimization step, we fix the pre-trained conditional mean estimator, giving us this X hat, and replicate its architecture with one key change at the output. We change the number of the images to be K, okay, to accommodate K principal components. And then this output is fed into a Gram-Schmidt procedure that maintains their orthogonality by construction, right? So the network is outputting orthogonal principal components. Now, next we feed this network with the predicted posterior mean and the input image. And once it outputs the top principal components, we learn these by injecting these alongside the error from the source image into a PCA objective that tries to maximize the variance along the predicted components, right? So we're projecting the error from the source image and we're trying to maximize the variance along the directions we find. Now, in the interest of time, I did skip a lot of details, obviously. For example, one trick that we found very useful in order to ensure that the principal components are actually aligned with the principal components and we're not only recovering the principal component subspace up to some rotation, was this stop-grad trick we introduced in the Gram-Schmidt. And we can also, in the paper, we have a way also to output the variances along the different components, so you know how much to move along each one. And we, as I said earlier, we can train this entire orchestra with one model. It takes the input image and output both the mean and the principal components. Okay, so this seems like a lot, so let's validate this on a 2D example. So for that, we tested this simple setting of Gaussian denoising, where we're assuming we have some IID Gaussian noise added to some measurement that comes from a GMM prior, a Gaussian mixture prior. In this case of 2 Gaussians. So here you see, for example, samples from this prior distribution, and on top, with each, connected with each, you see the red dot, which is basically the noisy measurement of each sample. And this basically produces the training set that we train our method on. So now after training, we wanted to test this method. So because it's a toy example, you can calculate the analytical posterior for every test point, right? So in this case, we have 2 test points, y1 and y2, and on red, in the red heat map, you see the analytical posterior being depicted on top of the prior, which we can calculate in a closed form. And here you see also the ground truth, principal components, and variances rendered as analytics. So in this case, again, derived from the analytical posterior. And now you see in black our method in both test points, and you see that in both cases, our method recovers the underlying ground truth very accurately. And we also have in the paper similar results in high dimensions, in 100 dimensions, for example. You're welcome to look there. Okay, so moving on to some image examples. Next, we tested our method on the task of m-less digits denoising. So this is the input measurement, right? You see it's very noisy. And this is the predicted mean in this case. And these are the top 5 principal components that our method predicts. So if we move along the first principal component, for example, you see it's changing from a 4 to a 9, which is basically very intuitive given the measurement. You see that the pixels closing the gap here are really uncertain, really noisy. And some other principal components also depicted digit style variation. So it's a 4, but all kinds of 4s in this case. And similarly for the rest. Okay, so we also tested this method on other data sets. For example, on the Cerebral HQ data set for the task of inpainting. So in this case, the mouth area is covered. And this is the posterior mean and top predicted principal components. And you see, for example, in this case, it's manipulating mouth opening or closing, sorry. And also for super resolution, the principal component in this case, if you look at the measurement, the eyebrow position is really not certain. So it basically depicts all kinds of versions of raised or lowered and eye opening or closing. And also on colorization, you see that the top principal component basically changes the background color, which makes a lot of sense. Okay, moving on to an application where this is actually important. So obviously I'm skipping here some tables with some quantitative numbers where we do well, but please just believe me for now and let's move on to something more interesting. So a very interesting application for such uncertainty models is, for example, biological image-to-image translation. And so the idea here is basically you want to take some biological sample and to predict how this sample would have looked like if you would have introduced different types of fluorescent dyes, right, and imaged it afterwards. And why this is very important, because if you can actually have good models of this type of task, then you can take some biological sample, you can predict all the different compartments of this cell or all the different subcellular elements in the configurations, and then in the overlaid volume, we can put everything into context. Right, and this is very important. Context is very important in biology, and why not simply do this in the experiment? Why do we need a model to do this? Because the more dyes, the more fluorescent tags you add into a sample, it starts to die slowly. So this increases phototoxicity, and therefore, this basically can enable a much prolonged observation window, right, and live cell imaging process. Okay, specifically the task that I will show you in a minute that we applied our method to was this basically dye-to-dye transfer, okay, where the task here is to take the image of a biological sample, image with one fluorescent dye, and transfer it to appear as if it was imaged with another. So the output, we're actually fluorescently imaging the cells, and the input, we're fluorescently imaging the cellular metrics, which are these so fluids in between cells. and the task is to go from left to right. So if we apply our method to this task, this is the input measurement that you see here on the top, and this is the predicted posterior mean, and these are the top principal components. So in this task, we have some prior knowledge, so we can actually make sense of the resulting principal components. For example, the first one basically tells a very known fact in this field is that we can't actually measure contrast perfectly. You can't recover the exact intensity of pixels with this task. And also the second one basically depicts that there are some cells being added or removed, so they're not certain in the reconstruction. It's not certain that there's a cell there. And also the third one tries to change the cellular shape. So for example, here it's trying to elongate it into an ellipse or to make it more roundish. And actually, these things are known in the field to be useful properties. So for example, this is the source image that generated that input measurement. In this case, it was measured jointly with it. And you can see that, for example, in the middle, the principal component actually predicted correctly that this cell is wrong. This cell shouldn't be in the reconstruction. And this affects cell counting. And also on the right, you see that the second principal component predicted that this cell should be more elongated, which also affects roundness measures. So, for example, in sickle cell anemia, cells tend to be more elongated or more roundish, and this has meaning. Okay, so moving on to multimodal posterior. So this seems like principal components can solve everything, but not exactly, right? For example, one very known limitation of principal components is that it cannot handle non-continuous multimodal posteriors. Right? So, for example, look at the glass option, at the glass example here. It's either you have glasses or you don't have glasses. You can't have somewhere in between. There's no like shades of glasses. So in this case, we need to do something smarter and different. Right? It seems we need to take a different approach. So one idea that one might think of from classical theory is to do clustering, right? So if you have distinct, discrete options, what you can do is to cluster the different options into different clusters. And then, for example, to present the uncertainty, you can simply report the cluster mean, right? For different clusters, with glasses, without glasses, or where this average is averaging a lot of different glass options. And in addition to this cluster mean, we can also report the relative cluster size, basically telling us how likely is a certain cluster versus not. So what we're essentially proposing now from a statistical point of view is to take the posterior mean and decompose it into its underlying constituents, where each one is accompanied by its likelihood, right? Okay, so two clusters in this case don't depict much uncertainty, it's either glasses or not, but we would like to get some finer details in this case. And so you might think, okay, just increase K, have 4 clusters instead of 2. Well, this is a problem because what tends to happen in K-means is that we focus on these high-density nodes and start segmenting them further and further, neglecting the low-density ones. And a better solution, actually, in this case, is to take the already constructed 2 clusters and further partition or recluster the samples in each one to subclusters, right? Predicting basically a tree in this case, or representing the uncertainty in the shape of a tree. This tree has several actually benefits. For example, it also enables faster user exploration. So assuming you're only interested in glasses, you don't need to visit this entire option, right? You just go directly to the glasses and then think which one you're interested in visualizing. And this also highlights rare cases, which is a very big plus because usually the problem with posterior sampling and eyeballing lots and lots of samples is that the rare case you're looking for is somewhere in between. You need to really skim through a lot of them to basically nullify your suspicion about the signal. All of this is nice, but this requires posterior sampling, right? And the entire talk, I am trying to convince you that this is a bad thing because it's very slow. So this is exactly what we did in our follow-up work, is doing this entire thing just faster. So how can you do this faster? And this brings me to the last point in this talk. Okay, so in this case, we want to take some input measurement and basically using a neural network in a single form pass, output the entire posterior tree, decomposing the mean into its underlying constituents. At this point, you might look at the slide and say, okay, how is this going? How is this going to happen? And the idea is the following. In our method, the raw outputs of the network is first the tree leaves at the bottom. So we're not outputting the tree structure. We output only the bottom layer. and we output also their likelihoods. And then using these, basically using weighted averaging, we construct the tree from the bottom up, right? Until we end up with the tree root, which is what we want to be the posterior mean. How do we train this thing? The training algorithm is rather simple. We know the source image that generated this input measurement. So we take the source image, now pass it through our tree, comparing it each time to the closest prediction. So we start by the root, and then at the next level, we compare it to the cluster that's closest to it, adding to our loss function another MSE loss. And next, we only compare it within the branch to the closest leaf, right? So the idea is basically, we're in a sense enforcing the hierarchy structure in our training. Now, obviously, there's deeper statistical meanings to these things that I'm skipping, and there's also some details that I didn't really tell you here. For example, the one thing that we needed is to weight the different loss functions, so there's some betas going on. And I've also skipped a bunch of other optimization-related tricks. Okay, so how does this perform? Here you see the input measurement on the left, again in MS digits, and we're trying to do it in painting. And this is the resulting posterior mean, so it's some convoluted digit. And if we apply this tree at the first level, you see, for example, it's confusing a 3 and a 5. So it's decomposing it to the different 3s and a 5. And if we apply this further again, then you see it's refining each cluster and doing the subcluster. So it's different types of 3s and different types of 5s. And this obviously works also for other data sets, for example, mouth in painting in Celebra HQ. So again, in this case, this is the posterior mean, and this is the decomposed tree. And as you can see in the middle, for example, the mouth is being opened, while on the sides, the mouth is more closed. If I actually zoom in on these side clusters, then you see that basically, in this case, the chin is more roundish, and in this case, the chin is more square, and being refined further. And finally, obviously, we came back to the bio stuff because that's where stuff really interests us, or me at least. And this is the input measurement in this case. And if we input this into the network, this is the posterior mean and its decomposition. And what you can see in the posterior mean is that there's some cell here that it's not sure about. It's somehow dim. And if we focus on the two clusters with 95% of the probability, what you can see is basically that this cell appears on the right cluster and doesn't appear on the left. And comparing this back to the source image, you see that indeed this cell is not existent in the source image, which is with a higher probability, the cluster with a higher probability, and therefore it's actually correctly predicting a higher likelihood there. And you can also see that in the subcluster of this cell, there's yet another cell that's uncertain. Furthermore, the last thing here is that if you look at this cell right here, and the source image is more round, and here the subcluster is refining it to become either more elongated or more round. With that, I finished my two parts, which are very long. So some thanks are in place before we finish. Obviously, in a decomposition fashion, I owe a lot of things to Tomer and Yoav. Both were great academic fathers, each in his own style. And I learned much from you, obviously, and it was really a joy working with both of you on a weekly basis, interchangeably, depending on the project. And of course, a big thank you to all the CPL lab, which we like to call it privately the TIRAS group. And obviously to Yoav's nanobio optics group, which a lot of you are here. You obviously helped immensely in this journey. And obviously also some thanks to the CPL engineers. You guys were a beacon of light in times of need. And of course, I need to especially thank everybody that I worked on and played a key role in my research in some of my papers. Got a lot of help, most recently from Rotem, who's somewhere. Yeah, Rotem. And obviously, we need to thank the sponsors who gave us a lot of money in different settings. And finally, thanking you. So I would like to thank you in an experimental fashion yet again, in this case, it's a measurement. That's it. Thank you. Any questions?